Imagine a new respiratory virus is spreading. Laboratories need a test now, and there is one huge problem. They do not even have the virus itself to work with.
Wait, if you do not have the virus, how can you build a test for it? I would assume you need a real sample first.
That is the obvious assumption. But public health laboratories may need to identify suspected cases before routine access to virus isolates is available.
So the challenge is not just scientific accuracy, it is speed under uncertainty.
Exactly. The outbreak was showing signs of being broader than first thought, and international spread through travelers was already occurring.
Then waiting for the usual laboratory materials could become a serious bottleneck.
That is why this paper matters. Its title is Detection of 2019 Novel Coronavirus, 2019 nCoV, by Real-Time RT-PCR.
So the central question is almost paradoxical. Can you create a reliable molecular diagnostic test for a newly emerged virus before you have the virus material itself?
Yes. The researchers aimed to build and deploy a robust diagnostic workflow for public health laboratories despite that constraint.
So they were trying to turn genetic information into something laboratories could use immediately.
Right. This paper is not about treatment or predicting severity. It is about detecting the new virus when the outbreak is moving faster than traditional laboratory preparation. A diagnostic test has to get two things right. It must detect the target, and it must avoid confusing that target with other respiratory viruses.
Especially because different respiratory infections can look similar from the outside.
Exactly. A molecular test should reliably recognize the genetic material associated with 2019 nCoV, while remaining exclusive enough that other respiratory viruses do not trigger the same result.
And this new virus was closely related genetically to SARS coronavirus.
That relationship was useful for designing the workflow, but it also created a discrimination problem. A useful assay had to distinguish 2019 nCoV from SARS-CoV, rather than merely detect a broad coronavirus signal.
So the researchers were using similarity as a starting point while still demanding specificity.
Exactly. The genetic relationship helped them design the test, but the diagnostic goal required the final workflow to separate the new virus from its close relative.
Then the research question becomes concrete. Can a test built without virus isolates still detect the new virus reliably and reject the wrong respiratory targets?
Yes, and this was intended for public health laboratory settings, not just a single experimental demonstration.
So success meant more than getting one positive signal. It meant creating a workflow that could be validated and deployed across laboratories.
The core tool was real-time RT-PCR, a method used to detect RNA targets through a sequence of molecular steps.
Give me the short version, without turning this into a biochemistry lecture.
The RT part means reverse transcription. Viral RNA is converted into DNA. PCR then amplifies selected genetic sequences, and real-time means that amplification can be monitored as the reaction proceeds.
So if the selected sequence is present, the reaction can produce a detectable amplification signal.
Exactly. The clever part was designing the workflow before virus isolates were available. The researchers relied on the close genetic relationship between 2019 nCoV and SARS-CoV and used synthetic nucleic acid technology.
So laboratory-made nucleic acid material helped bridge the gap until actual virus material was available.
That is the basic logic. They could develop and validate the diagnostic approach without waiting for an isolate of the new virus.
But a synthetic target cannot show whether ordinary respiratory infections might accidentally trigger the assay.
Right. The researchers coordinated across academic and public laboratories and evaluated assay exclusivity using 297 original clinical specimens containing a full spectrum of human respiratory viruses.
That comparison matters because the test must answer both questions. Can I detect the intended target? And do other respiratory viruses create the wrong signal?
Exactly. They also designed the workflow to discriminate 2019 nCoV from SARS-CoV, addressing a particularly close genetic alternative. The main result was that the workflow reliably detected 2019 nCoV and could further discriminate it from SARS-CoV.
So this was not merely a broad coronavirus detector. The workflow could identify the new virus while separating it from a closely related one.
Correct. The exclusivity testing added another important piece. Across 297 original clinical specimens containing a broad spectrum of human respiratory viruses, the coordinated evaluation supported the assay's exclusivity.
That matters in public health laboratories, where respiratory samples can involve many different viruses.
Exactly. Those specimens tested whether clinically relevant alternatives would interfere with the intended detection.
What about laboratories outside the original research groups? How could they work with the method?
The study reports that control material was made available through the European Virus Archive Global, or EVAG, a European Union infrastructure project.
So the work included not just designing the assay, but supporting other laboratories that needed control material.
Yes, that connects to the author's broader conclusion. The study demonstrated the response capacity produced by coordination between academic and public laboratories in national and European research networks.
So the achievement was both technical and organizational, a diagnostic workflow created under severe constraints, then validated through a wider laboratory network.
The strongest lesson is not simply that RT-PCR can detect a coronavirus. It is that diagnostic development can begin before the most obvious resource, the virus isolate itself, is available.
That changes the sequence I would have imagined. First obtain the virus, then build the test, then distribute it.
This study shows another path. When genetic relatedness is known and synthetic nucleic acid technology is available, researchers can design a molecular workflow earlier and test whether it distinguishes the intended target from close relatives and other respiratory viruses.
And the network matters because a diagnostic method only becomes broadly useful when laboratories can validate it, obtain controls, and put it into practice.
Exactly. The paper treats scientific preparation and institutional coordination as parts of the same response.
So the practical takeaway is not skip validation because speed matters. It is almost the opposite. Use available genetic tools to start earlier, then validate carefully against realistic alternatives.
That is a fair reading. The researchers used synthetic material, comparison with SARS-CoV-2, and 297 clinical specimens containing other human respiratory viruses to build confidence in the workflow.
Which means the surprising part is not simply that they moved fast, it is that they found a way to move early while still asking whether the test was detecting the right thing.
And that is the enduring insight. Outbreak preparedness is not only having a test already on the shelf, it is having the methods, control materials, and collaborative networks that let a reliable test be created when the pathogen itself is still difficult to access.